Hello friends, I draw your attention towards the right side where we have a group of learned and esteemed speakers. They have been invited to discuss our topic, which is the demand of time. The term population explosion is of global concern. To cater the needs of every individual in this ever-increasing population, biologists, geneticists are finding out ways and means to increase the quality and quantity of food. But how this can be achieved will be highlighted by our speakers. So, let me introduce them to you. On the rightmost end is Mrs. C. Chaudhary from KV Panagar, then Mr. Ram from KV Singtam, Mr. S. Kushwaha from KV Sitapur, Mrs. Mridu, KV Manori, Mr. Manoj, KV Okha, and Mr. U. S. Yadav, NTPC Rehan. Whenever we pick up newspapers, there are news items or articles on plant breeding techniques. Mr. Manoj, can you please tell us something about plant breeding techniques?
Thank you, Mrs. Sawan, for asking this question. Plant breeding technique is the requirement of today's scenario, because as population increases, everybody needs food, everybody needs sufficient nutrition. So it is a technique, or we can say purposeful manipulation of plant species in order to create varieties or types of plants which are better suited for cultivation, give better yield, and are plant disease resistant. So nowadays it is very beneficial for creating new varieties with disease resistant ability.
Why is this technique advantageous?
Yeah, of course, it is very advantageous. When we talk about the diseases, plant diseases, so we every year we face a lot of losses due to plant diseases. So we can create new varieties having resistant to particular kind of diseases. They are also pest resistant, and they have better quality of seeds. So for creating such types of varieties, we use these plant breeding techniques.
Okay, thank you. Just now a term explant was mentioned. Now I request Mr. Kushwaha to tell us how a new plant can be had from these explants. Is there any technique? If yes, please tell us the name and explain it.
Thank you, Mr. Saha. As your question is concerned about the explant, we know that luckily plant cells have a special ability that they can be grown into mature plant. This ability of the plant is called totipotency. And by using this ability of the plants, we can grow new plants from the plant parts, cells or tissues. In this method, what happens? We take some cells or tissue from explant and put it into the test tube or culture plate in sterilized condition, totally sterile condition in well-equipped laboratory. By adding some nutrient media, that media should contain some carbon source. So that maybe sucrose some inorganic salt may be added, amino acids, and by adding growth regulators like genes and cytokinins. Especially genes are essential for the growth of root and cytokinins for the shoot system. So when they are added, the callus is formed, and from that callus, root and shoots can be developed. In this way, we can produce lots of plantlets from X plants. And producing lots of plantlets from X plant is also known as micro-propagation. So, this technique is also helpful in the biotechnology. When scientists they develop GM crops are GM plants, they develop only 1 or 2 GM plants, but
excuse me, what do you mean to say by GM?
GM plant, okay, Mrs. Sir. GM plants are the plants whose genetic material has been altered.
Okay.
Some foreign genetic materials have been introduced in the plants according to the requirement. So these GM plants are produced in laboratory, 1 or 2 GM plants are produced, and scientists, if they want to grow more plants at a time from that such GM plants, they can take some tissues from such GM plants and grow lots of plants at a time, and these plantlets can be distributed or can be provided to the farmers or in their research field to grow more plants at a time. So these techniques are essential in these fields.
Okay, so this seems
This
to
technique
be...
is tissue culture. This technique is also known as tissue culture.
Okay, so this seems to be a boon for the farmers. Well, we have discussed a lot about the enhanced food production in terms of plants. We have yet another group, that is animals, which provide us meat, eggs, milk. So let us explore the animal world. Mrs. Mridu, you have a vast knowledge in this field. The other day, you were telling that many experiments have been done with cattle. Tell us how advantageous it is, or is there any disadvantage also?
Definitely there are advantages, like there is increase in fertility and even productivity due to accumulation of superior genes in the progeny. There is disadvantage also, like inbreeding depression in inbreeding, where mating is done between animals belonging to the same breed, and if this inbreeding continues for a long time, especially close inbreeding, it leads to reduction in fertility and even productivity, and this we call inbreeding depression.
Okay. So you have been mentioning about inbreeding. Then is there any term like outbreeding?
Yes, outbreeding is also there.
What is this?
Outbreeding is between animals which are not related.
Okay.
Now there are 3 categories in this, where animals they are unrelated, but they should not share common ancestor for 4 to 6 generations. Now this method is called outcrossing, and if the breeding is between unrelated animals which belong to 2 different breeds, then this is crossbreeding, and if the unrelated animals they belong to 2 different species, then it is called interspecific hybridization.
Okay, thank you, brother. What I could understand is that these techniques have really increased both quality and quantity of food. Now, one thought is continuously pondering in my mind. Like, if we have a control over the mating between the animals of our interest, will it give a better response? Mr. Ram?
Thank you for asking question. The technique is artificial insemination. In this technique, semen of desired bull is collected and injected to the heated cow. And if it is not instant use, it is stored in a frozen condition. So this method we can do this technique.
Okay. So, you want to share something else regarding this? So can you tell us how the control methods can be achieved and what are its advantages?
As I told, in this method, the semen is collected and administered to the genital tract of heated cow, and cow gets fertilized. There are some advantages of this technique. In this technique, first advantage is there is no need of all breeder to keep bull. Number 2, there is no wastage of semen. Number 3, it can be transported from one place to distant place. So this, and another more advantage is we can get it from desired breed when we need. These are the advantages of this technique.
Okay. Well, Mr. U.S. Yadav, please share your views. Yes. I know you have quite a bit of knowledge in this field. When we talk of improved food quality, we often hear of a term biofortification. Can you throw some light on it?
Yes, ma'am. Actually, biofortification refers to breeding of crops to produce high level of vitamins and minerals or proteins and higher fat. It is the most practical means to improve public health. Biofortification, aim of biofortification is to increase the protein content and quality, iron content and quality, vitamins content, micronutrients and mineral content.
Okay. So just now I have received a message from the audience on my handset. It is regarding single cell protein. Madam C. Chaudhary, Mrs. C. Chaudhary, please tell us something about single cell protein and is there any advantage of SCP?
Well, you see, the conventional methods of agriculture, they are not sufficient. The conventional methods of agriculture, they are not sufficient to meet the nutritional requirement of the ever-increasing population of our country. So to meet this requirement, we are making use of microbes. So microbes can be grown on a large scale, commercial scale, and they can supplement the nutrients. So one very common example is spirulina. It is an algae which can be easily grown on wastewater of the potato processing plant, or on molasses, or on even straw, or even sewage.
Okay.
So in this way, there is less burden on the natural resources, as well as they can produce food enriched with vitamins, minerals, proteins, etc.
Okay. Madam, I missed the example. Could you please repeat the example?
It's spirulina.
And what's the spelling, ma'am?
Spirulin.
Oh, thank you. Well, coming back to Mr. Yadav, I'm sorry, I missed to ask one thing. Is biofortification of any importance in Indian scenario?
Definitely, madam. Indian Agricultural Research Institute, New Delhi, has developed several vegetables crops which are rich in vitamins and minerals. Vitamin A with carrot, spinach, pumpkin. Vitamin C in rich bathua, mustard, tomato, and iron and calcium rich bathua and spinach. Protein enrichment, beans and pea.
Okay, again coming back to Mrs. Mridu. Can you give some examples of outbreeding?
Yes, there is an example of outcrossing that is of Israel, which is a breed of sheep developed by Punjab, in which Bikaner heels they are crossed with merino rams.
Okay.
And there is another example of interspecific hybridization of mule, where a female donkey is crossed with a male horse.
Okay. Well, what I feel is Mr. Kushwaha is very anxious to again share something in this topic. Mr. Kushwaha, can you please tell how can we produce virus-free plants from virus-infected plants?
Very nice question, Mrs. Shah. In case of epidemics, when total cropland in a particular area, total cropland is affected by the virus, then the farmers cannot grow new plants by using the seeds and other plant parts. So that time they can be helped by using the tissue culture technique. As we know that apical and axillary meristems of the plants, they are free of virus. If it is infected with the virus, then the cells or tissue which can be taken from the apical or axillary meristem and they can be grown artificially in the laboratory, and in this way we can develop so many plantlets by using tissue culture techniques, and we can help the farmer to produce the large number of plants in absence of seed which are already infected with the virus, because the virus infected plants, if they produce seed and other plant parts, they may be infected with the virus, so they should not use such type of seed. So in case of such epidemics, the scientists or laboratories which are near by that locality, they can help them by using the tissue culture techniques.
Okay. So this discussion has really proved very beneficial, at least for me. I have been inculcated with various terms like tissue culture, inbreeding, single cell protein, and whatnot. Well, now I would like to draw attention of my viewers. If you have any question to put up, so please come forward, and panel people are ready to answer. Yes, ma'am, please.
My question is for Radhu ma'am. Ma'am, how can one overcome inbreeding depression?
Inbreeding depression can be overcome by single outcross. If inbreeding depression becomes a problem, then the selected members of the breeding population should be crossed with unrelated superior animals, but they should belong to the same breed. This can restore fertility and even productivity.
Okay, thank you ma'am.
Are there any more questions?
Yes.
Okay, madam, please come. Thank you for being a part of our discussion.
I want to know from Mrs. Chandra Chaudhary whether there is any other microbes that can also help in the food production besides spirulina.
Well, an interesting question. Yes, there are several microbes. I would like to explain it in this way. Say, for example, you have a cow. It's a big animal. Say a cow weighing 250 kgs can produce protein, say about 200 grams. But say there is one microbe called Methylophilus methylotropus. Only 250 grams of this microbe can produce about 25 tons. If I am not mistaken, it can produce about 25 tons of protein in the same period. So you can imagine to what extent these microbes can be put into use to meet the requirement of the nutrients in the growing population. Besides this, there are several other microbes like mushrooms and all. They are also gaining importance in our diet today. So several varieties of microbes are there.
Okay, thank you.
Thank you, ma'am. So at the end, I would like to thank our panel of expert speakers. They have really thrown light in short, but they have really done their best to explain these scientific terms which will definitely enhance the food production, whether it is the case of plants or animals. And of course, as I said earlier, it has also been extremely beneficial for me in person. Thank you.